Okay, so we are now done discussing the synthetic function of the liver. So for this particular video, I will be discussing to you the excretory and the secretory functions of the liver. Okay, so the liver is capable of forming and excreting bile and bilirubin. Okay, so bile is actually very useful for the degradation of fat. It helps in the digestion process. And bilirubin is the principal pigment of bile, which is derived from the breakdown of the red blood cells. Okay, so if for a normal red blood cell, okay, it has actually a lifespan of 120 days. Okay, so that is the lifespan of a normal red blood cell. So after 120 days, these red blood cells will undergo degradation or hemolysis, okay, which leads to the release of hemoglobin, leading to the production of bilirubin. Okay. So after the degradation of a mature red blood cell, so once a mature red blood cell reaches its lifespan of 120 days, it will undergo in the process of degradation, which will lead to the release of hemoglobin. Now, the heme portion of your hemoglobin will be converted into bilirubin. Okay, and there are two different types of bilirubin. Okay, so one is we refer to as the conjugated or indirect form of bilirubin. Okay, another one is the conjugated, okay, or the direct form of bilirubin. Okay, so the conversion of the unconjugated to the conjugated form of bilirubin is possible due to the presence of an enzyme known as your UDPGT or your uridyl diphosphate glucuronyl transferase. Okay, so once the unconjugated bilirubin, okay, will be converted now to conjugated bilirubin, okay, the other name for conjugated bilirubin is your bilirubin diglucuronide. Okay, so that you can clearly understand the process of hemoglobin degradation leading to the production of bilirubin and urobilinogen, I have here a diagram. So kindly follow me, guys, as we discuss or as we go through the process of the formation of bilirubin. So again, immature red blood cell, okay, has a lifespan of 120 days. So, gitagaan ra siya na mulive og 120 days in our circulation. Now, after 120 days, okay, this will now become old red blood cells. And these old red blood cells will go to the reticuloendothelial system. Okay, reticuloendothelial system. And in the reticuloendothelial system, we have your phagocytic cells. Okay, now this phagocytic cell, okay, will eat or will engulf the red blood cell. Once the red blood cell is eaten by the phagocytic cell, it will undergo degradation or lysis. So there is a disruption of the RBC membrane. So once the RBC membrane has been disrupted, it will lead to the release of hemoglobin. Hemoglobin, okay, inside the phagocytic cell will be divided or there will be a breakdown into two parts, okay? One is the heme and one is globin, okay? So we have the heme portion and the globin portion. So the globin, okay, will be reused in the body, okay, and it will be stored in the amino acid pool, okay? For the heme, okay, a part of the heme, okay, will release iron and iron will be converted to its storage form in the name of ferritin, and it will be used in the development of premature red blood cells or younger red blood cell. Okay, and a part of the heme portion, with the help of the enzyme heme oxygenase, it will lead to the conformation of biliverdin. Okay, biliverdin will be converted to bilirubin. Okay, and once biliverdin is now converted to bilirubin, it will now release, okay, towards or to the plasma. So again, the type of bilirubin, okay, that is being converted from your biliverdin is your bilirubin 1, okay, or B1. This type of bilirubin is your unconjugated indirect form of bilirubin. Okay? Now, once your bilirubin 1 is in the circulation, okay, it will bound to the protein albumin. Okay? So your unconjugated bilirubin will bound to your protein albumin. Now, albumin with your bilirubin 1, okay, will go to the liver. Okay, so it's your albumin who will transport your bilirubin 1 to the liver. Okay, now inside the liver, your bilirubin 1 will be converted to bilirubin diglucuronide with the help of the enzyme glucuronil transferase. Okay, so bilirubin plus glucuronic acid, okay, will form bilirubin diglucuronide. Now, this form of bilirubin will be classified as your bilirubin 2. Okay, bilirubin 2. Your bilirubin 2 is again conjugated, and it's the direct form of bilirubin. Okay, so this type of bilirubin is being classified as conjugated because this bilirubin is being conjugated or being attached by your glucuronic acid. Okay, so that's why it's the name of this particular bilirubin is bilirubin diglucuronide. Okay, so it is attached to your glucuronic acid. Now, after the conversion of uh, bilirubin 1 to bilirubin 2, your bilirubin 2 or your bilirubin diglucuronide will go, okay, from the liver, will pass through the bile ducts leading to the intestines. Okay, so inside the intestines, we have bacteria, right? So our intestines, okay, have have normal flora, or our intestine is inhabited with bacteria. Okay, uh, so you have your bacteria inside your intestine, and the bacterial enzymes will reduce your bilirubin 2 to stercobilinogen and urobilinogen. Okay, so the byproduct of the metabolism of bilirubin 2 are your urobilinogen and stercobilinogen. So small amounts of your urobilinogen will enter the enterohepatic circulation leading to the kidney. So a little amounts of urobilinogen will be normally released through the urine, and some will be used again in the formation of bilirubin 2. Moreover, in the aspect of your stercobilinogen, it will be converted to stercobilin, okay, leading to the pigment, okay, leading to the formation of the pigment, which is responsible for the brown color of your stool. So stercobilinogen, it will be converted to stercobilin. On the other hand, those amount of urobilinogen inside the intestine will be converted to fecal urobilin. Okay, so your fecal urobilin also is a pigment responsible for the brown color of your feces or stool. So that's it for the hemoglobin degradation process and the production of bilirubin and urobilinogen. Okay, so I have here a table which will compare the two major types of bilirubin. Okay, so we have your bilirubin 1 and your bilirubin 2. Okay, so again, your bilirubin 1 is being referred to as the unconjugated form of bilirubin. On the other hand, your bilirubin 2 is the conjugated one because it is attached or it is bind to your glucuronic acid. Okay, aside from that, your bilirubin 1 is being classified as water insoluble bilirubin. That is why we don't find traces of bilirubin 1 in the urine. On the other hand, bilirubin 2 is water soluble. That is why we are using the chemical reagent strip. Okay, for bilirubin, okay, the one that we are identifying is in the form of bilirubin 2. Aside from that, your bilirubin 1 is also known as your indirect uh, reacting bilirubin, and for bilirubin 2, direct reacting. Okay, so then your bilirubin 1 will increase during prehepatic disorders. Okay, so when you talk about prehepatic, from the word pre, which means before, so these are the conditions related, or these are the diseases, okay, related or that happen before the liver. So example of this, okay, you have your intravascular hemolysis. Okay, when you talk about hepatic, okay, so these are the conditions, okay, that happens within the liver. So different liver infections such as hepatitis or um, uh, hepatitis or cirrhosis. Okay, and when we talk about post-hepatic disorders, these are the disorders that happen outside the liver, post, okay, after the liver. So example of these are the formation of gallstones and obstruction of the bile duct. So we will discuss all of these disorders in our next topic. Okay, so again, guys, as a review, the product, okay, of the metabolism of the bacteria in relation to your bilirubin, to the conjugated form of bilirubin, is your urobilinogen and stercobilinogen. Okay, so again, guys, some amounts of urobilinogen will enter the hepatic circulation, okay, leading to leading to your kidney, and the kidney will excrete amounts of urobilinogen, and some also will be used in the formation of conjugated bilirubin. Okay, and for stercobilinogen, okay, stercobilinogen cannot be reabsorbed, okay, and it remains in the intestine wherein it is oxidized to your stercobilin. So remember, guys, the product of the oxidation of your stercobilinogen is your stercobilin. On the other hand, those amount of urobilinogen that is in the intestine, okay, or when it's recirculated, okay, or reaches in the intestine, it will oxidize, okay, it will undergo oxidation, and the product will be your urobilin. So urobilin is the oxidation product of your urobilinogen, and stercobilin is the oxidation product of stercobilinogen. So the common denominator among these two pigments is that these two pigments are the ones who is responsible for the characteristic brown color of feces. That's why ang normal color sa atong stool or feces, guys, kay brown, light brown, or dark brown due to the pigments of stercobilin and urobilin. So let's now discuss the other function of the liver, and that is detoxification and drug metabolism. Now, these are very important functions of the liver because it can allow important substances to reach the systemic circulation and can serve as a barrier to prevent toxic or harmful substances from reaching the systemic circulation. So the body has two mechanisms for detoxification of foreign materials, which includes your drugs and poisons, and metabolic products, which includes your bilirubin and ammonia. Now, every substance that is absorbed in the gastrointestinal tract again must first pass in the liver, and this is being referred to as the first pass. Okay, the first pass. So it's the liver. The liver is the organ responsible for the detoxification of the toxin, and of course the delivery of the drug, okay, to the different parts of the body. So the most important mechanism for uh, drug metabolism is that the liver is responsible for the detoxification of a lot of drugs through different metabolic processes. So we have oxidation, reduction, hydrolysis, hydroxylation, carboxylation, and demethylation. Okay, so we all whenever gani guys that we take drugs, we have to make sure Jude, that we are taking the right dosage of the drug, okay, or we follow the proper schedule whenever we take medications, because again the liver is the front line organ for the detoxification process. So if there is inaccuracies, okay, in your drug intake, it can actually affect the function of the liver. Okay, so after we discuss the main functions of the liver, let's go now to another accessory organ. So we have your pancreas. So the pancreas is a unique organ because it has two major function, okay? Or this particular organ can be classified into two. It can be classified as an endocrine tissue or an exocrine tissue. So when you talk about an endocrine organ or tissue, it is responsible for the release of hormones, right? So inside your pancreas is you have your islets of Langerhans. Okay, so your islets of Langerhans is the one responsible for the secretion of hormones such as insulin, glucagon, gastrin, and somatostatin. Okay, so we all know that insulin regulates the glucose levels in our body. This is actually a hypoglycemic agent. Okay, so in times when there is high increase in glucose in our body, your insulin is the one who counteract that, and your insulin is the one who decreases the blood glucose. Okay, so what the insulin does is that the hormone insulin, okay, facilitates the penetration of glucose, okay, from the blood circulation to the cells, okay, to the different cells of the body, leading to the decrease of blood glucose in the circulation. On the other hand, your glucagon, gastrin, and somatostatin are what we called as your hyperglycemic agent. So hyperglycemic. So these particular hormones will increase or will facilitate the release of glucose to from the different cells or tissue into or towards the circulation. Okay, so that is one of the function of your pancreas. So another function of your pancreas is the release of enzymes, okay, digestive enzymes, okay, which helps in the breakdown of food. Okay, so the normal protein rich pancreatic fluid is actually clear, colorless, and watery. So the pH of a pancreatic fluid can reach up to 8.3. Now, the alkalinity, by the way, of the pancreatic fluid is caused by a high concentration of sodium bicarbonate, okay, which is present in the pancreatic fluid. So, which is used to neutralize the hydrochloric acid in the gastric fluid from the stomach as it enters to the duodenum. So for the pancreatic fluid, okay, so we have different types of uh, digestive enzyme that is present in your pancreatic fluid. So we have the proteolytic enzymes. Okay, so we have different types of enzymes under the proteolytic enzyme. We also have the lipid digesting enzymes. That's number two, the lipid digesting enzymes. We also have the carbohydrate splitting pancreatic amylase and your nucleases, or being classified as your ribonucleases. Okay, so which is also responsible for the degradation of sugars or carbohydrates. Another accessory organ is your gallbladder. So usually the gallbladder is a pear-shaped structured located under beneath the liver and at the right side of your abdomen. So the primary function of your gallbladder is of course the storage of bile. Okay, so remember guys, okay, gallbladder is not the organ responsible for the production of bile. It's the liver. But the gallbladder is the organ uh, which stores the bile. Okay, so whenever the bile is not used for digestion, the storage or the reservoir for the storage of bile is your gallbladder. So the main function of your bile helps in the digestive process, especially in the breaking up of fats. Okay, breaking up of fats. So it also drains the waste products from the liver into the duodenum and some part of the small intestine. So please go to the next presentation for the different laboratory procedures or tests in relation to the accessory organs of the digestive system.